inheritance and variation. Living beings produce offsprings of their own kind. The young ones produced by sexual reproduction always resemble their parents and also each other. But still, we show considerable variation. What is inheritance? It is a process by which characters or traits are passed from parents to their progeny. That is, from one generation to the next. It forms the basis of hereditary. What is variation then? It is a degree by which the progenies differ from their parents. Genetics is a branch of biology which deals with the inheritance as well as the variation of the characters from parents to their offspring. In this chapter, we will study about these similarities and variations in characters and also their inheritance pattern from one generation to other. Mendel's law of inheritance. It was during mid-19th century that headway was made in an understanding of inheritance. Graham Johann Mendel conducted experiments for the first time to understand the pattern of inheritance and variations in living things. His observations became the foundation of modern genetics and study of hereditary. Therefore, he is also known as father of genetics. He conducted hybridization experiments on garden pea for 7 years and proposed the law of inheritance in living organisms. Mendel conducted artificial and also cross-pollination experiments by performing emasculation on several true breeding pea lines. A true breeding line refers to one that have undergone continuous self-pollination and show stable trait inheritance and expression for several generations. Mendel's experimental approach. Mendel's methodology during the investigation of the inheritance pattern included mathematical logic and statistical analysis of the data. His experiments had a large sampling size that gave greater credibility to the data that he collected. The confirmation of the inferences from his experiments on successive generation of his test plants proved that his results pointed to the general rules of inheritance. Mendel's experimental material. He selected garden pea plant as his experimental material due to the following reasons. Pea plants are readily available on large scale and has bisexual clistogamous flowers. Peas are self-pollinated and can also be cross-pollinated. These are annual plants with short life cycle, so several generations can be studied within a short period. Pea plants differ in distinct and contrasting characters which provide many easily detectable contrasting characters. Pea plants can be easily raised, maintained and handled also. Mendel selected 14 true breeding pea plant varieties as pairs which were similar except for one character with contrasting traits. Mendel's observation and interferences. Mendel observed one trait or character at a time. Example, he crossed the tall and the dwarf plant to study the inheritance of one gene. He hybridized plants with alternate forms of a single trait. The seeds produced by these crosses were grown to develop into plants of filial F1 progeny or F1 generation or F1 plant. Then self-pollinated the tall F1 plants to produce plants of filial 2 progeny or F2 generation. In later experiments, Mendel also crossed pea plants with two contrasting characters known as dihybrid cross. Observation. In F1 generation, Mendel found that all the pea plants were tall and none of them were dwarf. He also observed other pair of traits and found that F1 plant always resembled either one of its parent and the trait of the other parent was not found. In F2 generation, he found that some of the offsprings were dwarf. That is, the characters which were not seen in F1 generation were expressed in F2 generation. The proportion of plants that were dwarf were 1/4 of the F2 plants, while 3/4 of the F2 plants were tall. These contrasting characters, that is tall and dwarf, did not show any mixing either in F1 or in F2 generation, as none of that was intermediate height. Similar results were obtained with the other traits that he studied. Only one of the parental traits was expressed in F1 generation, while at F2 generation stage, both the traits were expressed in the ratio of 3:1. Mendel also found identical results in dihybrid cross as in monohybrid cross. Mendel's interferences. He proposed that some factors pass down from parent to offspring through the gametes over a successive generations. Nowadays, these factors are known as genes. Genes are hence the units of inheritance. Genes which code for a pair of contrasting traits are known as alleles or allelomorphs. That is, they are slightly different forms of the same gene. Gene occurs in pairs in which one dominates the other, called as dominant factor, or the gene which expresses itself while the other remains hidden, and it's called as recessive factor. Allele can be similar in case of homozygous and dissimilar in the case of heterozygous. In a true breeding tall or dwarf pea plant, the allelic pair of genes for height are identical or homozygous. Capital TT and small tt are called the genotype of the plant, while the characters tall and dwarf are called as phenotype. When tall and dwarf plants produce gametes, by process of meiosis, the alleles of the parental pair segregate and only one of the allele gets transmitted to a gamete. Thus, there is only 50% chance of a gamete containing either allele, as segregation is a random process. During fertilization, two alleles, that is capital T from one parent and small t from other parent, are united to produce a zygote that has one caps T and one small t, or the hybrids of capital T and small t. Since these hybrids contain alleles which express contrasting traits, the plants are heterozygous. Punnett square. The production of gametes by the parents, the formation of zygotes, the F1 and F2 generations can be explained by a diagram called as Punnett square. It was developed by a British geneticist, Reginald C. Punnett. It is a graphical representation to calculate the probability of all possible genotypes of offsprings in a genetic cross. The ratio of 1/4:1/2:1/4 of capital TT is to capital T and small t is to both small t is the expression of A plus B the whole square, that has the gametes bearing genes in an equal frequency of 1/2. The expression is 1/2t plus 1/2 small t the whole square is equal to, if you expand this formula, you'll get 1/4 capital T plus 1/2 capital T and small t plus 1/4 small t. Though the genotypic ratios can be calculated using mathematical probability, but it is not possible to know the genotypic composition by looking at the phenotype of a dominant trait. Therefore, to solve this problem, Mendel devised test cross, back cross and test cross. Back cross is a cross of F1 progeny back to one of their parents. A special back cross to a recessive parent is known as test cross. This method was devised by Mendel to determine whether the dominant phenotype is a homozygous or heterozygous. There can be two possible genotypes of an unknown dominant phenotype as illustrated. If the unknown is homozygous violet, then crossing with white recessive gives all violet offsprings. If the unknown is heterozygous, crossing with white recessive results in violet and the white progeny in the ratio 1:1. In the case of double heterozygous, that is heterozygous violet and axial flower crossed with double recessive white and terminal flower, and the rest cross ratio will be 1:1:1:1. One gene inheritance. The study of inheritance of a single pair of alleles or factors of a trait at a time is called as one gene inheritance. Based on his observations on monohybrid crosses, Mendel proposed two general rules in order to consolidate his understanding of inheritance in a monohybrid cross. Today, these rules are called as principles or laws of inheritance. The first law is law of dominance, and it is also the first law. Characters are controlled by discrete units called as factors. Factors occurs in pairs only. In a dissimilar pairs of factor, one member of the pair is dominant while the other is recessive. This law is used to explain the expression of only one of the parental characters in a monohybrid cross in F1 generation and the expression of both in the F2 generation. It also explains the proportion of 3:1 ratio obtained in the F2 generation. Law of segregation is known as second law. This law states that though the parents contain two alleles during gamete formation, the factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors. Hence, alleles do not show any blending and both the characters are recovered as such in the F2 generation, though one of these is not seen in the F1 generation. Two genes inheritance. To verify his results of monohybrid cross, Mendel also crossed pea plants differing in two characters, that is dihybrid cross. This helped him to understand the inheritance of two genes at a time. It was found that inheritance of one pair of allele does not interfere in the inheritance of the other pair of allele. Based upon this, Mendel proposed a second set of generalizations or postulates which is called as law of independent assortment, which is known as third law. This law states that when two pairs of the traits are obtained in a hybrid, segregation of one pair of characters is independent of the other pair of characters at the time of gamete formation. It also gets randomly rearranged in the offsprings, producing both parental and new combinations of characters. Deviations from Mendelism. All the patterns of inheritance could not be explained exclusively on the basis of Mendel's original principles alone, and certain complexities were observed by later workers which deviated from Mendel's law or Mendelism. The first one is the incomplete dominance. It is a phenomenon in which the phenotype of F1 hybrid offspring does not resemble any of the parent, but it is an intermediate between the expression of two alleles in the homozygous state. Here, the phenotypic ratio deviated from Mendel's monohybrid ratio, but the parental characters reappear in F2 generation. For example, in snapdragon and 4 o'clock plant, there are two types of flower color in pure state. On crossing the pure breeding red flowered and white flowered plant, hybrid of F1 generation possesses pink color. And on selfing the F1 plants, F2 generations comes as one red and two pink and one white flowered plant. Here, pink color is due to incomplete dominance of red flower trait over the white flower trait. And the next is codominance. It is a phenomenon by which two alleles express themselves independently when present together in an organism. In other words, it is a phenomenon in which offspring show resemblance to both the parents. Example, ABO blood grouping in humans. ABO blood groups are controlled by a gene I. The plasma membrane of red blood cells has sugar polymers that protrude from its surface, and the kind of a sugar is controlled by the gene. The gene I has three alleles, IA, IB and small I. The alleles of IA and IB produce a slightly different form of the sugar, while allele small I does not produce any sugar. In humans, each person possesses any two of the three I gene alleles. IA and IB are completely dominant over small I. When IB and small I are present, only IB expresses because I do not have any sugar. Same in the case with IA with small I. But when IA and IB are present together, they both express their own types of sugar. This is due to the codominance. Therefore, the red blood cells have both AB types of sugar. Since there are three different types of alleles, there can be 6 different combinations. Multiple alleles. When more than 2 alternative forms of a gene occupy the same locus on a chromosome in a population, these are known as multiple alleles. The ABO blood grouping is a good example of multiple alleles. In this case, more than 2, that is 3 alleles are present governing the same character. Multiple alleles can be found only when population studies are made. Pleiotropy. It is a phenomenon in which a single gene product may produce multiple or more than one phenotypic effect. For example, phenylketonuria is a disorder caused by mutation in the gene coding the enzyme phenylalanine hydroxylase. The affected individual show hair and skin pigmentation and mental retardation. In garden pea, starch synthesizes size of a starch grains and the shape of seeds is controlled by one gene. The genotypes and phenotypes for the traits are capital B indicates round seeds and large starch grains. Capital B and small b indicates round seeds, intermediate size starch grains. Small b indicate wrinkled seeds and small starch grains. Therefore, it can be said that dominance is not an autonomous feature of the gene or its product, but it depends on the production of a particular phenotype from the gene product. Polygenic inheritance. It was given by Galton in 1833. In this, traits are controlled by 3 or more genes. These traits are called as polygenic traits. The phenotype show participation of each allele and is also influenced by the environment and is called as quantitative inheritance, as a character or phenotype can be quantified. For example, human skin color, which is caused by a pigment melanin. The quantity of melanin is due to the 3 pairs of the proteins A, B and C. If a black or very dark and a white or very light individuals marry each other, the offspring shows intermediate color, which is also called as mulatto. A total of 8 allele combinations are possible in gametes, forming 27 distinct genotypes, complementary genes. It is a phenomenon when 2 pairs of non-allelic dominant genes which interact to produce only one phenotypic trait, but neither of them produces the trait in the absence of the other. It shows the phenotypic ratio 9:7. Example, production of flower color in sweet pea, that is lathrice. Rediscovery of Mendel's law. Though Mendel published his work on inheritance of characters in 1865, it remained unrecognized for several reasons till 1900. In 1900, de Veres, Correns and von Schirchmark rediscovered Mendel's results independently. Due to microscopic, they carefully observed cell division. This led to the discovery of chromosomes. Molecular basis of inheritance. Mendel suggested that there are some factors or genes which help to maintain the phenotypes and genotypes of organisms after generation. After years of investigation of various scientists, it came to the realization that DNA is the genetic material in majority of organisms, while RNA acts as a genetic material in some viruses but has additional roles as well. It functions as adapter, structural in some cases as a catalytic molecule. In this chapter, we will discuss the structure of DNA, its replication, the transcription process, the genetic code and their regulation. In the last section, the genomics and the essential of human genome sequencing and its consequences will also be discussed. The DNA and the RNA world. Genetic material is that substance which controls the inheritance of trait from one generation to other. Following are the criteria that a molecule must fulfill to act as a genetic material. It should be able to replicate itself. It should be stable both chemically and structurally. It should provide the scope for slow changes which are required for evolution. It should be able to express itself in the form of Mendelian characters. According to these criteria, both DNA and RNA have the ability to direct their duplication using specific processes, whereas the other molecules in the living system fail to fulfill the first criteria itself. Example, proteins. Search for genetic material. The discovery of nuclein by Miescher and the proposition of principle of inheritance by Mendel were almost at the same time, that is 1869 to 1866 respectively. But the fact that DNA acts as a genetic material took a long time to be discovered and proven. By 1926, the quest to determine the mechanism for genetic inheritance had reached the molecular level, and gradually the question, what molecule acts as a genetic material, got answered. Transforming principle. Frederick Grith in 1929 carried out a series of experiments with Streptococcus pneumoniae. He observed that when these bacteria were grown in a culture plate, some of them produced smooth, shiny colonies named as S-type, whereas the others produced rough colonies named as R-type. This difference in appearance of colonies, that is smooth and rough, is due to the presence or absence of mucus polysaccharide, which is caught on S strains but not on R strains. In this experiments, he first infected two separate groups of mice. The mice that were infected with the S strain died from pneumonia, as S strains are virulent strains causing pneumonia. So the S strain was injected into a mice, the mice died. The mice that were infected with R strain thus did not develop pneumonia and they lived. In the next set of experiments, Griffith killed the bacteria by heating them. The mice that were injected with heat-killed S strain bacteria did not die and lived. Whereas on injecting a mixture of heat-killed S strain and live R strain bacteria, the mice died. Moreover, living S bacteria was recovered from the dead mice. From all these observations, Griffith concluded that the live R strain bacteria had been transformed by the heat-killed S-strain bacteria. That is, some transforming principles had transferred from the heat-killed S-strain, which helped the R-strain bacteria to synthesize a smooth polysaccharide coat and thus become virulent. This must be due to the transfer of the genetic material. However, he was not able to define the biochemical nature of genetic material from his experiments. Biochemical characterization of transforming principle. Oswald Avery, Colin MacLeod and Macklin McCarthy worked to determine the biochemical nature of transforming principle in Griffith's experiment in vitro system. Prior to this experiment, the genetic material was thought to be a protein. During this experiment, purified biochemicals from the heat-killed S cells were taken to observe which biochemicals could transform live R cells into S cells. They discovered that DNA alone from heat-killed S-type bacteria caused the transformation of non-virulent R-type bacteria into S-type virulent bacteria. They also discovered that protein digesting enzymes and RNA digesting enzymes did not inhibit this transformation. This proved that the transforming substances was neither protein nor RNA. DNA digesting enzyme caused inhibition of transformation, which suggests that the DNA caused the transformation. Thus, these scientists came to the conclusion that DNA is the hereditary material, but still not all biologists were convinced. HERSHEY-CHASE experiment. The proof that DNA is a genetic material came from the experiments of Alfred Hershey and Martha Chase in 1952. They carried out some experiments with viruses that infect bacteria called as bacteriophages. The genetic material of bacteriophage enters the bacterial cell after the bacteriophage gets attached to the bacteria. The bacterial cell treats the genetic material of the virus like its own genetic material and then produces more virus particles. Hershey and Chase experimented to find out whether it was protein or DNA from the virus that entered into the bacteria. They took two separate media for growing these bacteriophages. Out of two, one medium contained radioactive phosphorus and the other medium contained radioactive sulfur. Viruses are then grown on each medium. The virus grown in the presence of radioactive phosphorus contained radioactive DNA but not radioactive protein. This is because DNA contains phosphorus and not protein. In the same way, the viruses grown in the medium containing radioactive sulfur now contained radioactive protein, not the radioactive DNA. This is because DNA does not contain sulfur. These radioactive viruses were then allowed to attach to bacteria. As the process of infection with virus continued, the bacteria were agitated in a blender and the viral coats were removed from the bacteria. When they were spinned in a centrifuge, the virus particles separated from the bacteria. They observed that the bacteria that were infected with viruses containing radioactive DNA were radioactive, whereas the bacteria that were infected with radioactive proteins were not radioactive. This indicates that only DNA entered the bacterial cell from the viruses. Thus, the genetic material that is passed from the virus to bacteria is DNA. From the Hershey's and Chase experiment, the fact was established that DNA acts as a genetic material. But later studies revealed that in some viruses, like example tobacco mosaic virus, QB bacteriophage, etc., the RNA is the genetic material, the DNA. Deoxyribonucleic acid acts as a genetic material in almost organisms. It is a long polymer of deoxyribonucleotide. The number of nucleotides or base pairs present in the DNA determines its length. This length is also the characteristic of an organism. Example, in E. coli, there is 4.6 into 10 to the power 6 base pairs. In human beings, there is 3.3 into 10 to the power 9 base pairs. Structure of a polynucleotide chain. A nucleotide is composed of nitrogenous base, a pentose sugar and a phosphate group. A pentose sugar, two types of sugars are present. That is ribose in the case of RNA and deoxyribose in the case of DNA. Nitrogenous structure. It is a nitrogen containing organic molecule having similar physical properties of a base. There are two types of nitrogenous bases. Purines, that is adenine and guanine, pyrimidines, thiamine, uracil and cytosine. Out of pyrimid, cytosine is common for both RNA and DNA, while thiamine is present in DNA and uracil is present in RNA. A nucleoside is formed when a nitrogenous base is linked with the pentose sugar through a N-glycosidic linkage. Example, adenine plus deoxyribose sugar gives deoxyadenosine. Guanine plus ribose sugar gives guanosine. Guanine plus deoxyribose sugar gives deoxyguanosine. A phosphate group. A nucleotide is formed when a phosphate group is linked to a 5' OH of a nucleoside through a phosphoester linkage. Two nucleotides, when linked through a 3' to 5', a phosphodiester linkage form a dinucleotide. In a similar fashion, more nucleotides may join to form a polynucleotide chain. The polymer chain thus formed has one end with a free phosphate moiety of ribose sugar. This is marked as 5' end of the polynucleotide chain, and then the other end with a free hydroxyl group marked as a 3' end of the polynucleotide chain. Thus, the sugar and the phosphate form the backbone in a polymer chain, and the nitrogenous bases linked to sugar moiety project from this backbone. In RNA, there is an additional OH group at 2' position in the ribose of every nucleotide residue. Discoveries related to structure and composition of DNA. Friedrich Miescher identified DNA as an acidic substance present in nucleus. He termed it nuclein. This compost was later renamed as nucleic acid due to its acidic properties by Altman in 1899. Levin in 1990 found DNA to contain phosphoric acid as well as deoxyribose sugar. He characterized 4 types of nucleotides present in DNA. Erwin Chargaff in 1950 formulated important generalization on the basis and other contents of DNA called as Chargaff's rule. It states that for a double-stranded DNA, the ratio between adenosine and thiamine and guanine and cytosine are constant and equal to 1. That is, adenine plus thiamine by guanine plus cytosine gives 1. James Watson and Francis Crick in 1953 proposed the double helix model for the DNA structure on the basis of X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin in the same year. One of the important features of this model was the complementary base pairing. It means if the sequence of bases in one strand is known, the sequence in other strand can be easily predicted. Also, if each strand from a DNA acts as a template for synthesis of a new strand, the daughter DNA thus produced would be identical to the parental DNA molecule. Watson and Crick model of DNA. Watson and Crick worked out the first correct double helix model of DNA, which explained most of its properties. The salient features of double helix structure of DNA are: the DNA is made up of 2 polynucleotide chain and the backbone is constituted by a sugar phosphate, while the heterogeneous bases project inward. The 2 chains have antiparallel polarity. That is when One chain has 3' to 5' polarity and the other has 5' to 3' polarity. The bases in two strands are paired through hydrogen bonds to form the base pairs. Adenine is bonded with thymine of the opposite strand with the help of two hydrogen bonds. Guanine is bonded with cytosine of the opposite strand with the help of three hydrogen bonds. So, a purine bonds with a pyrimidine always, thus maintaining a uniform distance between the two strands of the helix. The two polypeptide chains are coiled in a right-handed fashion. Pitch of the helix is 3.4 nanometer or 3.4 into 10 to the power minus 9. The number of base pairs in each turn is 10. Distance between a base pair in a helix is 0.34 nanometer. Percentage calculation of bases is done by adenine plus thymine is equals to 100 minus guanine plus cytosine. The plane on one base pair stacks over the other in double helix. This provides the stability to the helical structure in addition to H bond. The length of DNA in E. coli is 1.6 nanometer while in humans it is 2.2 millimeter. Packaging of DNA helix. The length of DNA in humans can be calculated as distance between two consecutive base pairs is 0.34 nanometers equals to 0.4 into 10 to the power minus 9 meter. So the total number of base pairs in a DNA helix in a typical mammalian cell is equals to 6.6 into 10 to the power 9. Therefore, the length of this DNA helix would be 2.2 millimeter approximately. Thus, we see that the length of the DNA is far greater than the dimension of a typical nucleus, which is approximately 10 to the power minus 6 m in a diameter. Therefore, there must be some special arrangement to accommodate such a long polymer in a cell of both prokaryotes and eukaryotes. In prokaryotes such as E. coli, a well-defined nucleus is absent. So, DNA is held together with some proteins in a region known as nucleoid. The DNA in nucleoid is organized in large loops held by proteins. In eukaryotes, the organization of DNA is much more complex in eukaryotes. Depending upon the abundance of amino acid residue with charged side chains, a protein acquires its charge. Histones are proteins that are rich in basic amino acids, that is lysine and arginine. Both of these amino acids carry positive charges on their side chain, so histones are set of positively charged basic proteins. These histones organize themselves to make a unit of 8 molecules known as histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer, forming a structure known as nucleosome. There are 5 types of histones. DNA connecting the two adjacent nucleosomes is called as the linker DNA. One nucleosome contains approximately 200 base pairs of DNA helix. Nucleosomes are the repeating units of chromatin, which is a thread-like stained bodies present in nucleus. The nucleosomes in chromatin look like beads on string when observed under a microscope. The chromatin is packed to form a solenoid structure of 30 nanometer diameter, and further supercoiling tends to form a looped structure called as chromatin fibers and then the chromatin. This further coils and condenses at metaphase stage of cell division to form the chromosomes. Non-histone chromosomal protein. These are additional sets of proteins that contribute to the packaging of chromatin at a higher level. These regions present in chromatin are euchromatin and heterochromatin. Euchromatin are the regions of chromatin which are loosely packed in a typical nucleus. Heterochromatin are the regions of chromatin which are densely packed. Euchromatin is the transcriptionally active chromatin, while heterochromatin is transcriptionally inactive chromatin. RNA world. Ribonucleic acid was the first genetic material discovered. It is evident through various scientists' researches that essential life processes such as metabolism, translation, and splicing, etc. evolved around RNA. RNA used to act as a genetic material as well as catalyst, but RNA, being a catalyst, was reactive and hence unstable. Therefore, DNA has evolved from RNA with chemical modification that makes it more stable. DNA being double-stranded and having complementary strands further resists changes by evolving a process of repair. A nitrogen bases in RNA are of two types, purines and pyrimidines. Purines include adenosine and guanine. Pyrimidines include cytosine and uracil. Ribosugar plus nitrogenous base gives ribonucleoside. Ribonucleoside plus phosphate group gives ribonucleotide. Central dogma.